Good evening, everybody, and welcome to our event here at Princeton Public Library in our virtual space on Crowdcast. This event tonight is co-sponsored by MIT Press and Labyrinth Books. And I put information in the chat for you to let you know that Labyrinth Books is providing a 10% discount to anybody who buys the book. This discount will last for a week. You can order online at labyrinthbooks.com, or you can email them at orders@labyrinth@gmail.com, or you can just call them. Their phone number is all there. And if you're ordering online, there is a promo code for you to use, which is just Shaw, one of the author's last names, to let them know that you were here at the program tonight. So please remember, it's important to support independent local bookstores, and we're so fortunate to work with Labyrinth, providing series of events here, both on the library's platform on Crowdcast as well as on Labyrinth's platform. They're also here on Crowdcast, and we call this series Library and Labyrinth live streams. Just a few housekeeping items. If you're having any problems with the sound or the video, you can there's a button here at the bottom. It says get audio or video help. You can try that, or you can put something in the chat. I will close the chat down for the portion of the program until we get to Q&A. But if you have a question that you want to ask the authors, there is an ask a question button. Just pop it in there. And again, right here, there's a direct link if you want to purchase the book from Labyrinth. So I think that's about all of the housekeeping, and I see lots of people logging in. And so I think we're ready to get going with the actual portion of the talk. So we are really fortunate to have two experts here tonight in immunology with us to discuss their new book that interweaves explanations of scientific principles and ongoing efforts to combat COVID-19 with stories of the people behind the science. And I was just talking to the authors in the green room, and they say this is a pandemic project. They started working on it in March, and they worked really hard to turn this around quickly. And they also, one of their goals was to provide an accessible explanation of how viruses emerge to cause pandemics, and then how our immune system combats them, and how diagnostic tests, vaccines, and antiviral therapies work. And so they're really trying to make this so that it's accessible to all, because these concepts are ones that provide the foundation for our public health policies. So in just a moment, I'll be bringing up our guest speakers. We have Replication Chakraborty, who is the Robert T. Haslam Professor of Chemical Engineering and Professor of Physics and Chemistry at MIT, where he also served as the founding director of the Institute for Medical Engineering and Science. He is a founding member of the Ragon Institute. And we also have Andre S. Shaw, an immunologist and staff scientist in immunology and oncology at Genentech, who holds adjunct professorships at Washington University in St. Louis and at the University of California, San Francisco. So we have them on different coasts of the country at the moment, but thanks to the magic of the internet, we can bring them both up here with us, coming in from San Francisco and coming in from Massachusetts. Welcome to the talk. I'm going to just disappear. I'll be here online with you, and we're going to get going. Take it away.
I'm just going to bring up my screen. So can everybody see my screen? I hope.
Yes.
Okay. Thank you very much, Janie, for the introduction. And thank you to we're very grateful to the Princeton Library for providing us with this opportunity to speak about our book. So maybe to get started, I'll say a few words about what motivated us to write this book. The human story is inextricably linked with infectious disease-causing pathogens and pandemics that they cause. But because of improved sanitation, vaccines, and antibiotics, we, the inhabitants of the 21st century, had largely forgotten about the human and economic toll that can be exacted by pandemics. So when COVID-19 hit, many of us did not have the concepts and facts that had suddenly become so pertinent. There was a deluge of information, but the information came in snippets rather than building the concepts layer by layer so that one could develop a holistic perspective. So we thought that a short book, which could develop such a perspective in an easy to understand way, may arm readers with the knowledge to understand not only what's going on today, but also think about pandemics potentially that could come in the future. And most importantly, enable them to be informed participants in the debates that will hopefully ensue about how we might create a more pandemic resilient world. And as Janie mentioned, we weave stories of the science with stories of the people behind the science on whose shoulders we have had to stand recently to, for example, make vaccines so rapidly, and on whose shoulders we will have to stand in the future to create a more pandemic resilient world. Now today we are going to try to briefly outline the topics that we cover in our book, and the illustrations and drawings that we will use to illustrate the ideas are all in the same informal style as in our book, and which are all the creation of Philip Stork, who is a scientist at Oregon's Health Science University. So as mentioned, the written record is replete with stories about great plagues and smallpox epidemics and so forth. Our ancestors noticed that those who recovered from disease could safely serve as caregivers. And the Chinese and Indians started to harness this observation to try and protect people from smallpox. Material from the scabs of smallpox patients would be collected, stored for a while, and then administered. This process came to be known as variolation. Variolation was brought to Europe, first to UK actually, and then to Europe, and by Mary Wortley Montagu. And that resulted in the first sort of organized clinical trial that was run by Caroline, then Princess of Wales. But you know, variolation was a very dangerous procedure. Many people who were variolated died, and often there were localized smallpox outbreaks. So variolation never became very popular and couldn't protect people from smallpox at scale. All of that had to wait till Edward Jenner found that by inoculating people with cowpox, a relatively harmless pathogen, one could confer protection against smallpox. And this discovery ultimately led to eradication of this scourge from the planet. But remarkably, Jenner did not know that infectious diseases were caused by microbial pathogens, let alone that we have an immune system. The discovery of that microbial pathogens cause infectious disease had to wait till the 19th century, when Pasteur and Robert Koch learned how to observe, isolate, and grow microbes and study their properties. And these studies led Pasteur, for example, to make the serendipitous discovery that you could inoculate people with dead or inactivated pathogens and still confer protection. The 20th century saw its share of pandemics, the polio epidemics, which led Salk and Sabin to develop vaccines. The 1918 influenza pandemic, which likely contributed towards ending the First World War, and then the HIV pandemic, which, although under control somewhat in most parts of the world because of antiviral therapies, we still don't have either a cure or a vaccine for HIV. In the 21st century, we have seen 3 epidemics and pandemics caused by the same family of pathogens, namely coronaviruses. Now, you know, when Pasteur was making his vaccine against rabies, he could not isolate and study its properties. And that's because virus particles which are such a threat to human life, are tiny, about 1/1000th the thickness of a human hair, and so they can't be seen with a standard microscope. Now, when a virus enters your body, it has to enter your cells, and it does so in the following way. Viruses have a spike made up of proteins, and for example, the spike of the virus SARS-CoV-2, which causes COVID-19, binds to a molecule on the surface of our cells called ACE2, which is involved in regulating blood pressure. ACE2 is found in cells abundantly in the lung, blood vessels, tissues involved in smell, heart, kidney, etc. So this virus can infect cells in many different tissues and potentially cause complications in several different organ systems. Now, once a virus binds to a molecule on our cell surface, it still has to force its way in. And the way SARS-CoV-2 does it is that it co-opts one of the molecules in our own cells, which serves like a scissor, to cut the spike into two, which makes it look like a spring has been released, and that action releases the contents of the virus into our own cells. Now the virus has to replicate, that is, make many copies of its own proteins so that many different virus particles can be assembled to go on to infect other cells. To see how this works, let's just remind ourselves about how we make copies of our own proteins. So in order to function, we need proteins, and a cell is the quantum functional unit in our body, if you like. And if you think of the cell as a car, proteins make up all its parts. So the cell has to make many copies of our proteins. And information about the proteins that we can make is encoded in DNA, which is stored in the nucleus. DNA, as you know, is a molecule made up of two strands. Each strand is composed of 4 units or bases, T, A, C, and G. And the reason the 2 strands wind around each other in the form of a helix is because T can only bind to A and C can only bind to G, and so the 2 strands are complements of each other. Now, the way information about our proteins is stored in DNA is the following. Proteins are made up of amino acids. We have 20 types of amino acids, and 3 triplets of letters on our DNA that code for different amino acids. So, for example, A, C, and G codes for amino acid 1, G, C, C for amino acid 2, and so forth. Now, how is this information translated to producing proteins? Happens in 2 steps. First, the DNA, the letters on DNA, is transcribed into another molecule, RNA, by a molecule called RNA polymerase. Now, RNA is an ancient molecule, also made up of 4 units, with the only difference with DNA being that now instead of T, you have a U. So this RNA polymerase makes many copies of the RNA, and then this RNA is translated into proteins, the corresponding protein, by a huge protein machine we have in us called the ribosome. And this 2-step process, DNA to RNA to protein, is called the central dogma of molecular biology. Now, viruses come in 2 broad categories, DNA viruses and RNA viruses. DNA viruses carry information about their proteins encoded in DNA. Examples include herpes viruses, chickenpox virus, and so forth. RNA viruses encode information about their proteins actually as RNA. Coronaviruses and influenza viruses are examples. But viruses cannot translate the information encoded in DNA and RNA into their own proteins and multiply on their own. For that, they have to infect plants, animals, or us, and then hijack our cells' machinery to make their own proteins and begin to multiply and make many copies of themselves. So viruses really are like experienced travelers who are always on the move, and so they pack light. They only carry the things with them that they can't find in the hotel, and that being our own cells. Now, the genome of DNA viruses is translated into its proteins by our cell's machinery as per the central dogma. RNA viruses break this rule. So, for example, viruses like coronaviruses bring with them a molecule called RNA-dependent RNA polymerase, which runs along the RNA genome of the RNA virus and makes another copy of the RNA, and they make many such copies of the RNA, and then these RNA copies are then translated into the viral proteins by our own ribosome. Now, HIV is an example of a retrovirus, and these guys are even weirder. They take their RNA, and first they convert that to a complementary DNA. This is done by a molecule called reverse transcriptase. This DNA corresponding to the virus's genome is then inserted into our cell's genome. And then, as per the central dogma, that DNA is converted to an RNA and then to a protein. Now, when the human genome was first sequenced, a surprise was that a significant fraction of the human genome was comprised of ancient retroviral genes that were silent. So apparently, we've been doing battle with retroviruses for a very long time. Now, so this is the life cycle of an RNA virus. The virus enters, makes lots of copies of its RNA. They're assembled into, they're then translated into proteins. Virus particles are assembled, they're released, and they go on to infect other cells. Now, antiviral therapies, like remdesivir, for example, try to block one or more of these steps in the viral replication cycle. Now, as I've mentioned, viruses are ancient organisms, and we've lived with them forever. But the kinds of viruses that infected our ancestors were probably different in both type and diversity compared to those that prevail today. Many of the viruses that circulate today, like the one that causes COVID-19 and flu, they are spread by casual contact between people. Some people died due to acuity of disease, but our immune system in most of us usually vanquishes this pathogen or virus, and then a memory of this infection is imprinted in our immune system so that upon reinfection, we can just swat the virus away without trouble. Such viruses would be very unlikely to survive in a pre-agrarian world. Our ancestors then lived in small communities, spread over large tracts of land, so an infected person encountered very few people in their daily lives, and so infected very few people. Now, if the virus was a lethal virus, then a highly lethal virus, then all it would do is it would kill off all the people in this little community, and then it would have no one to multiply in, and that viral species would go extinct. Now, if the virus could be vanquished by our immune system, then too, very soon, there would be no more susceptible people in this community, and the viral species would again have no one to multiply in and would go extinct. So the viruses that infected our ancestors were likely not terribly contagious or deadly, and our immune system was not successful in completely killing off these viruses. In fact, these viruses adapted to coexist with us and stayed silent most of the time, and periodically would rear their heads, and then our immune system, and multiply, and then our immune system would again suppress them, and this cycle would go on. DNA viruses, like herpes viruses, are examples of such ancient viruses that circulate in the human population today. Now, all of this changed when we learned how to grow crops, because we started living in larger communities, which were also more densely populated. And so an infected person could transmit the virus to many people. And In a large, dense population, new births and trading with nearby communities, which also existed now, provided a steady stream of newly susceptible people. We also started to domesticate animals. So viruses that typically circulated in animals only could now begin to infect us. These effects were exacerbated with population growth, the Industrial Revolution, which led to urbanization, and easy travel. So here are some snapshots of the density of human population in 1000 AD, when the population was 256 million, to 20 years ago, when the population was 6.4 billion. And you see that in addition to fast population growth, you see highly dense populations, and regions that were completely unoccupied are now occupied, which meant that we started to be in proximity with even newer kinds of animals. Thus, it came to be that a diverse set of viruses that can be easily spread by casual contact, such as those that cause these diseases, began to circulate in the human population. Now, how does a virus emerge to cause pandemics? Well, you know, these processes by which, the processes by which we copy our DNA into proteins is a very high fidelity process. But these processes that RNA viruses use are actually very error prone. So for example, when an RNA polymerase is copying the RNA of a coronavirus and it has to copy it into a U, it can make an error so that this U becomes a G. Reverse transcriptase makes similar errors in retroviruses. So now, if the RNA is different, then the protein that it will produce is also different, so the virus that will be assembled is also different. Now, most of these mutant strains of virus are not very functional, so they're worthless. But sometimes a mutation can emerge that not only is as fit as before, but can actually make this virus even more functional. For example, this is how the mutant strains of SARS-CoV-2 that were first isolated in the UK and South Africa that are more transmittable must have emerged. Now, viruses can also change guise in other ways. So imagine that in a cell, in an animal or a human, is infected by two viral strains, perhaps one from an animal and one from a human. Alternatively, you could imagine an animal like a bat, in which different strains of coronaviruses coexist. A cell in this bat, could get infected by two strains of the coronavirus. When that happens, the RNA polymerase that runs along, let's say, this red RNA, copying its RNA into other copies and then to proteins, can hop off this one and start copying the other one. As a consequence, the viral strain that will be produced is a recombinant strain, if you like, because it's a hybrid virus made up partly of the red proteins and partly of the blue proteins. Now, viruses like influenza carry their RNA genome as distinct segments, sort of cassettes, if you like, that can be easily swapped in and out. So if such a virus, two strains of such a virus infects a cell, then you see you can have a viral strain produced where some of the cassettes come from the red one and the others come from the blue one. This process is called reassortment. Now, viruses that circulate in animals, they have adapted to thrive in that animal. So they don't typically infect us or thrive in us because we are different. But sometimes, because of the malleability of the RNA genome, by mutation, recombination, and reassortment, a new viral strain can get produced that can now infect humans. Alternatively, a new viral strain can emerge in one animal that can infect now another animal. And in that animal, it could go through a few more mutations and become capable of infecting us. The viruses that cause SARS, MERS, COVID-19, and influenza pandemics undoubtedly emerged in this way. When such a new virus emerges, no one has immunity to this virus, and if it's reasonably contagious and reasonably lethal, frightening pandemics such as the ones we are going through can occur. But we have won most of our battles with such viruses, and we will win it against COVID-19 as well. And this is because we have an immune system which usually vanquishes these viruses, and we learned how to harness the immune system to make vaccines. And Andre will now describe these important topics.
So before I start, I should remind everybody that although I'm an employee of Genentech, anything I say today is really my views as a private citizen. So we know that the immune system has been evolving in concert with the evolution of the pathogens that are infecting us, and that we have been in constant battle with many pathogens over centuries, millennium, including viruses. What we've learned over the last 100 years is that there are two main parts of the immune system, and we call these parts the innate and the adaptive immune system. Most of us are familiar with the adaptive immune system because that's the part that makes antibodies and T cells. And these antibodies and T cells are generated to be very specific for a specific pathogen and are generally highly effective at getting rid of them. Because these weapons are organized and generated in a custom fashion, it's a relatively slow process to generate antibodies and T cells during an infection, and it usually takes about 1 to 2 weeks. So if we had to rely only on the innate immune system or the adaptive immune system to survive, it's so slow to react that the pathogen much of the time would have killed us before the adaptive immune system was ready. So we've learned in the last 30 or 40 years that there's a whole nother arm of the immune system that's called the innate immune system. And this part of the immune system functions to alert the body that a pathogen has entered. And it uses some broad one-size-fits-all mechanisms to basically buy time for the adaptive immune system to turn on. The innate immune system has specific receptors that can recognize viruses, bacteria, and fungi. And when it recognizes that something like that is in the body, it triggers a system-wide response that's mainly through the release of hormones that immunologists call cytokines. And these cytokines cause things that we're all very familiar with, like fever, like muscle ache, like fatigue, like lack of appetite, chills. These cytokines also change your metabolism, they change your blood flow, they increase your ability to clot. When you begin to feel sick, it's not the pathogen that's causing that. It's the innate immune system turning on. And so that's a sign that something has entered your body, that the innate immune system has recognized something. And it's basically trying to harness weapons to slow the growth of the pathogen and also tell the adaptive immune system that it needs to get started finding a specific weapon for the pathogen. So probably the easiest way to think about how the innate and adaptive immune system works is to think about it in the context of a COVID infection. So we know today that a person is infected exposed to the virus, and they don't generally feel sick for about 5 days. So what's happening during those 5 days is the virus is replicating, and at some point enough virus is made that the innate immune system can recognize it, and you start to feel sick. You have a cough, you've got fever, you have muscle ache. The adaptive immune system then starts the clock on the innate immune system, then starts the clock on getting the adaptive immune system going. And that, as I just said, takes about a week. And when it does come on, the adaptive immune system is able to clear the virus from the body. And as the viral levels are getting lower and lower, the signals to the innate immune system are starting to turn off, and the patient starts to feel better. And then the virus is eventually completely cleared from the body. Now, one of the mysteries of COVID is that it appears that you can infect people for a couple of days before you get sick. And this is telling us that there's some problem with the innate immune system recognizing COVID early enough. And these 2 days are giving the virus a kind of a head start. The virus can be growing and growing, and the innate immune system and the adaptive immune system are really not doing anything. And so this largely explains why COVID patients can infect others before they start to feel sick. Now, in severe cases, the innate immune system fires, the adaptive immune system fires, and probably in many cases, by the time the adaptive immune system fires, there's so much virus around that it's much harder to get rid of and get these viral levels to lower and to clear. And this has cascading detrimental effects because now the innate immune system cannot turn off. And the innate immune system was not designed to be on for weeks at a time. It was really designed to be on during this period between innate immunity and adaptive immunity. And this is very detrimental to the health of the patient. At this point, about 7 days after you start to feel sick, the adaptive immune system is not being able to decrease viral load and make you feel better, is when patients generally are sick enough to need to go to the hospital. In some cases are sent to the ICU. Most of our new medical treatments like steroids, the specialized drugs that dampen the innate immune response, the COVID antibodies, they all need to be given relatively early, and that's because we need to basically slow the virus growth during the early period so it doesn't get out of control. And this is to buy time for the adaptive immune system to clear the virus. So how does the adaptive immune system work? The virus enters the body, goes into the cell, as Arup has described. It hijacks the cellular machinery and starts to make millions of copies of itself. Eventually, all of these viruses are released from the cell into the blood. And there are two components of the adaptive immune system that we have that are there to get rid of it. The first is the B cell, and this is the cell that makes antibodies. There's another part of the immune system, the T cells, and that's there to identify the infected cells and get rid of them. Now, each of us, humans, we have over 10 billion different B cells, meaning each B cell has a completely different antibody. And so when a new virus enters the body, what we're trying to do is expose all 10 billion of these B cells to the virus, hoping that a few of our B cells will have an antibody that's specific to the virus. When this virus binds to the B cell that it has an antibody for, this will cause the B cell to divide and make many copies of itself, and also tell the B cell to make antibodies that it will secrete into the blood that can recognize the virus. And then through a process of Darwinian selection that we talk about in the book, these B cells have a process to make the antibody even better. And ultimately, hopefully, a really good antibody is produced by the B cell. And this can basically go around the body, bind to free virus particles in the blood, and remove them from the bloodstream. Now, antibodies are not very good at identifying cells that are infected with the virus that are producing the virions that are going out into the blood. And so to get rid of those cells, we have another arm of adaptive immunity called the T cell. Now, the T cell is not recognizing the free virion, but rather the cell that's making the viruses has a process where it takes small little pieces of the viral proteins and it throws them out on the surface. And it uses these little pieces of viral proteins on the surface as a flag, basically telling the T cells, I have something going on here. There's a protein that I've never seen before, and I'm putting it out on the surface. And in a process that's very similar to the B cell, we have over 10 billion different types of T cells. hopefully these T cells can recognize a vast array of different peptides. And what we're hoping during the adaptive immune process is that we find the T cells that are specific for the virus. And once they bind to the viral peptide on the surface of the cell, these T cells will proliferate, will make many copies, daughter cells of these. And then these cells will convert into specialized cells that function to get rid of the virus. And one of them is called the killer T cell. This cell, when it goes around and it recognizes this protein flag that's on the surface of an infected cell, it goes in there and it kills that cell. And eventually, the B cells making the antibodies, the T cells killing the infected cells, we basically get rid of the virus and we sterilize our bodies to this virus. So once the infection's over, we've made a lot of B cells, we've made a lot of T cells, and we don't need them anymore. And so during the process of clearance, we basically kill off most of the B cells and T cells that were generated. But what we do is we leave a few of them around. And these are called memory B cells and memory T cells. And this retention of these cells is to be prepared for the next time this virus enters the body. These T cells and B cells making the antibodies and recognizing the viral proteins are there. And they can basically instantly jump into action. They don't need to wait a week. They're already ready. They kill and clear the virus. And most of the time when this happens, you never notice. You go around your daily life, you never even realize that another virus had entered your body. So as we describe in the book, vaccines and vaccination function by stimulating an immune response and hopefully generating this memory response so that when you do get infected with the real pathogen, you're protected. So in the 17th and 18th centuries, Pasteur and Koch came up with two strategies to use as vaccine. One of them was the inactivated vaccine. So the idea here is I have a bacteria that I want to make a vaccine to, so I grow a whole bunch of it in the lab and I kill it, so it's dead. And then I take that dead bacteria and I inject it into you, and I hope that you'll generate an immune response to something in that dead bacteria, and that will give me memory, antibodies, and T cells that will protect me. And the best example of an inactivated vaccine or a dead vaccine that we use today is the influenza virus. Now, Pasteur also came up with another strategy, and that was the live attenuated strategy, where he would take a pathogen, and so he did this for rabies, and he would grow it repeatedly, hoping that he could weaken it to the point where when he injected it into a human, it wouldn't cause disease, but it would cause an immune response that would protect you with memory against future infections. And as we'll discuss in a second, the polio vaccine that Sabin designed is of this type, versus the polio vaccine that was designed by Salk, which was a dead vaccine. And for many of us, our children, they get the measles, mumps, and rubella vaccine, and that's another example of a live attenuated vaccine. In the 20th century, the big advance was generating the subunit vaccine. What we were trying to do in the 20th century is, I think a lot of people have problems getting injected with like dead bacteria or a dead virus or even a live attenuated virus. We know that the immune response is mainly directed against the proteins in the bacteria or the proteins in the virus. And so using modern recombinant DNA technology, the strategy was to basically use those methods to generate large amounts of recombinant viral proteins or recombinant bacterial proteins at industrial scale and use that material as vaccines. And it was much safer because you weren't actually using anything that had to do with the pathogen. You were using just the proteins. And good examples of these types of vaccines are the vaccines for papillomavirus, for hepatitis B, and then the famous diphtheria, pertussis, and tetanus toxoid vaccines. In the fight against COVID, we've been able to generate new 21st century strategies. The first strategy that made it to the market were the vaccines made by BioNTech, Pfizer, and Moderna. So as Arup has explained to you, RNA makes protein. And so rather in the subunit vaccine strategy of making bucket loads of protein, the idea was we could just inject the RNA encoding these proteins into the body. Cells would take up this RNA. They would use the instructions in that RNA to make COVID-19 proteins. And then you'd have immune response against that. And here, again, you're not getting the virus. You're just getting the instructions to make the proteins of the virus. In the second strategy, which is exemplified by the Oxford AstraZeneca vaccine and by the Harvard Johnson & Johnson vaccine, neither of these are approved yet in the United States, but we're expecting that they'll get approved very quickly. The idea here is to take a harmless virus that causes the common cold, engineer it so it has the DNA that encodes the proteins for the COVID-19 virus, inject these recombinant viruses into the body. These viruses will then release the DNA molecules. DNA will get made into RNA. RNA will get made into protein. And in this way, we generate the adaptive immune response. Should also remind us that the virus that is carrying these COVID-19 DNAs has actually been engineered, so it's a non-infectious virus. There's no way you can actually generate an infectious virus, and it's basically what we call replication incompetent. So one of the great pleasures of writing this book was to learn about the incredible story of the polio vaccine. I'm a little bit younger than the era of the polio vaccine, and so this was a story that I didn't really appreciate and realized writing the book that this is really one of the great scientific triumphs of the 20th century. Now, polio turns out to be a very, very interesting virus. There is very little evidence in the historical record for polio until the late 19th century. We first became aware of small clusters of children getting a childhood paralytic illness that was occurring about once a decade between 1870 and 1900, and only in Scandinavia and in the US. In the early 20th century, these epidemics continued to grow both in frequency and size, peaking in the United States in the 1940s of about 21,000 deaths per year, and that's not counting all the children that were becoming paralyzed. We learned that this disease appeared to mainly affect children younger than 10, and almost all cases occurred in the summer. Was this a new virus, or was this an old virus? If we look in the archaeological record, there's evidence that in the ancient Egyptians that there was a paralytic illness that might have been polio. What we currently think is that this is an ancient virus. It's been with us for thousands of years. And that prior to the 20th century, most children were exposed to polio during infancy. And during infancy, they rarely got sick, and that's probably because they were being breastfed. As hygiene got cleaner and cleaner, and as we started breastfeeding less and less, there was a delay in the exposure of children to polio, and they were now getting polio at an older age and were not being protected by mother's milk. And now they were getting more severe infections, and many of these were resulting in infection of the brain and spinal cord, causing paralysis. And this also explains why it was increasing mostly in developed countries. Understandably, there was a great fear for children in the summer months, and this resulted through the 20s, 30s, and 40s of closure of beaches, swimming pools, movie theaters, and any activities that included the gathering of young children. The infection of FDR when he was 39 was a galvanizing moment in the polio history, because he started a charity called the March of Dimes that was focused on developing a vaccine. In 1949, the first breakthrough occurred when John Enders at Harvard Medical School figured out how to grow the polio virus. The path for the development of a vaccine was clearly visualized by Jonas Salk, and he jumped on this finding. He figured out, as an academic physician, how to grow this in large quantity. He figured out how to inactivate it. And in 4 years, 1953, he was ready to test the safety of his vaccine on humans. He actually first tested it on his family. And then in 1954, after establishing safety, he was ready to do large clinical trials. 1954, we didn't know a lot about doing large clinical trials, and so there was a lot of controversy and discussion about how to do this. We knew that epidemics occurred in the summer, and so he knew that he needed to get his vaccine into a large number of children before the end of the school year. He knew that it infected young children smaller than 10, so he decided that the clinical population to test the vaccine on would be 7-year-old children. This was complicated by the fact that the Salk vaccine needed up to 4 or 5 boosters. And so all of this needed to get coordinated. They needed to make the vaccine. They needed to get into everybody. And then there were the controversies about who would the controls be? Would they use placebos? And in the end, they decided to use a couple of different trial designs, one of them placebo-controlled, but most of them not. And they enrolled 2 million children in the United States to be in the trial. At the end of the summer, they had to tabulate all the information. This was a huge trial, huge amount of data in a pre-computer age. It took almost 9 months to tabulate the data. And in the spring of 1955, it was announced with great emotion and excitement that we had a vaccine that could cure polio. And this was the Salk vaccine. Now, Albert Sabin was another polio scientist, 10 years older than Salk, and he had been working on polio since the 1930s. He was the one to realize that polio was spread by contaminated water and food and was coming into the body through the gastrointestinal tract. And this led him to believe that the best vaccine would be one that was swallowed and could protect the gastrointestinal tract. And so he focused on developing an attenuated strain of polio that didn't cause disease, that he could give to people by mouth. By the time he was ready, which is in the spring of 1955, he went to the March of Dimes in the US government and said, I have a vaccine that I would like to try. And the US government was like, we already have a vaccine. We don't need your vaccine. And so Sabin remarkably decided to go to the Soviet Union in 1955, in the middle of the Cold War. He convinced them to try and test his vaccine out. The Soviets generated a trial of over 12 million people, and their vaccine was eventually approved in 1961. It turned out that the Sabin vaccine was much easier to produce. It didn't require a syringe injection. It didn't require multiple doses. And people who got the Sabin vaccine could not get infected with the virus, in contrast to the Salk vaccine. The Sabin vaccine also conferred lifelong immunity. So it was used around the world, and it led to the eradication of polio in most parts of the world. In recent years, because the Sabin vaccine is a live virus, it did occasionally result in causing polio in a small number of individuals. And so as polio became less and less common in the world, the UK and the US governments decided that there was too much risk in using the Sabin vaccine. And so about 20 years ago, the US government went back to the Salk vaccine. I think the example shows how when we were generating these vaccines, we really had no idea what was going to work in the long term and be the best vaccine in the end. And what we've seen is that there were different periods of time where one was better than the other. We learned here that differences in the dosing, how long the immunity lasts, how easy it is to administer are all important issues, including the efficacy. And so today, when we argue about what's going on with the COVID-19 vaccines, we really don't know what's going to be the best vaccine in the long term. What is the best vaccine to use across the planet? And that we will see only in the next couple of years when we see what happens.
I'm going to come back and just take the last couple of minutes to comment briefly on how we might build more pandemic resilience. In our book, we talk about a number of ideas, and here, which all speak to how advances in science and technology could be leveraged to achieve a more pandemic resilient world. Here I'm going to talk only about two such examples, the first being testing. In many countries, when COVID-19 first emerged, one of the major problems in the early days was that we could not get and deploy a reliable testing system at large scale, and thus be able to identify, quarantine, and isolate people to prevent the spread of the disease. This is not surprising when a new virus emerges. And even now, our testing paradigm is one where you collect a sample, send it to the laboratory, and sometime later the test comes back. What we really need for the future are portable, reliable testing methods that can be deployed at scale in a point-of-use fashion and that can be rapidly adapted when a new viral virus emerges. This is not fantasy. Such things are being developed today. For example, this particular device developed by Collins and coworkers in the context of Zika. You collect a sample, you extract the virus's RNA, amplify it, and then synthetic biology is an approach where you can engineer bacterial cells to do what you want them to do. So they engineered a synthetic biology circuit that could detect the presence of the Zika RNA, and then once it did that, it secreted a colored chemical. This synthetic biology circuit was placed on top of a filter paper and then freeze-dried, and then you could transport it any way you wanted. And then at the point of use, you just put water and it worked. So if you put a dollop of a sample on it, then if it changed into this magenta color, it meant that you had the Zika virus. Now, this is the sort of tests that we need because they're easily adaptable. You just have to change that filter paper. So now let me finally close by speaking a little about the next stage of vaccines. So we've all heard that SARS-CoV-2 is mutating. And at least the South African strain, the current vaccines are less effective against it, but fortunately not hugely so. But now other mutations will emerge. So we're in a race as to who wins, the vaccination program or the emergence of new mutations. But on the other hand, you should look in the long run, when the whole world is vaccinated, we're going to put enormous selection pressure on this virus population to mutate to evade this vaccine-induced protection. So then different companies are going to make these different vaccine, as they're already doing that, but so it'll become like flu in a sense. But will that protect us from the next pandemic causing coronavirus? No. So maybe we should start thinking about pan-virus family vaccines. This again is not fantasy. Work done in the context of HIV and influenza has made a lot of progress, although has not achieved success yet, on creating such pan-virus family vaccines. Now, this is a depiction of the spike of the HIV virus. It has these yellow residues here that are conserved from one strain to the other. Influenza has a similar region. So if you can design a vaccination protocol that will target these regions with the immune response it generates, you're going to protect people from all strains of this. What we need to do is couple massive sequencing of viral strains, not only in humans of a particular family, but also global surveillance, including animals in which they circulate. And then take all that data and analyze it with the power of machine learning and modern computational models, and the incredible amount of, and combine that with the incredible amount of data we are collecting now on viral infections in the context of COVID-19, and discover what these targets ought to be and how we can generate vaccines that will target them with an immune response. These and other technologies will make us more pandemic resilient. The question is, will we make these investments so that the next time it is not the way it has been this time? With that, we'll stop. We apologize that we took a little long. We thank you for your attention, and we'd be delighted to take any questions.
Okay, so this is Janie here. I'm going to come back on screen. Hang on one moment. So I'm going to turn the chat back on in case anybody wants to put their question just in the chat. And I do see we have one question in the Ask a Question box. This has been extremely informative. I know I learned quite a bit sitting here watching and listening. And it is very interesting about the polio vaccine and the story and how that goes along. So, so somebody here has asked, can you be certain that COVID-19 wasn't made in a lab, since it spread so fast, mutated several times, become more transmissible, and appears to be more lethal and impacts so many parts of the body, not just the respiratory system? So, really, can we be certain that it was not manufactured?
Well, I'll start, and then Andre will I'm sure have more to say. One cannot be sure about anything with 100% certainty, but all the evidence that exists based on scientists analyzing sequences of the coronaviruses across animal species and across ones that have been in humans before and trying to trace the lineage of this particular virus have concluded that it was not made in a laboratory, but really all evidence points to the fact that it went through an intermediate animal and then jumped to us, from bats to an intermediate animal to us. And there are several pieces of evidence that suggest that that's what happened. Andre, would you like to add something to that?
I think it's a very interesting story. I mean, the parts of the story that are very compelling for people thinking that this is a man-made virus, and we tell a little bit about this story in the book, is that when SARS and MERS came out, we really didn't know where they came from. And so there was an incredibly dedicated Chinese scientist who lives in Wuhan, who took it as her own, personal quest to figure out where these viruses came from. And she was the one to discover that bats were carrying these organisms. And even then, she didn't stop. She wanted to make sure that she could find the original SARS and the MERS, the SARS virus, and she eventually did. And this involved her crawling around in caves all over the world, collecting feces from bats, analyzing it, collecting coronaviruses. And so she is the world's expert on coronaviruses, and she happens to live in Wuhan. So that is the part of the story where people, get hung up on. The evidence from looking at the sequence of the virus, there's really no evidence that that this could be engineered. Originally we thought it was from pangolin, but there are a lot of good molecular explanations for the origin of this virus. Now, part of this question is how it can infect all these different organs, but actually the ACE2 receptor is actually the same receptor that's used by SARS. And so the ability of this virus to infect ACE2 has been there for a while in a class of coronaviruses. SARS is much more lethal than SARS-CoV-2. So if you really wanted to generate something lethal, you would have picked SARS, not SARS-CoV-2. SARS-CoV-2 is got that kind of sweet spot where it's not completely lethal. It's pretty infective. I mean, I personally think that if it was 3 times more lethal or 5 times more lethal than it is, the whole country would be paying attention to this. It has a lethality that's just kind of on the borderline that can make it controversial. And so I don't see this as the perfect virus that somebody would want to engineer.
On the other hand, just to make one final remark, if you have such a lethal virus like SARS was, it's actually easier to control because these people fall sick and you can isolate them more easily. But anyway, that's...
I mean, it's something we thought a lot about when Arup and I investigated. We talked to a lot of experts. We really kind of thought this through. And I think Occam's razor, you know, the simplest explanation is that this was generated, you know, through a natural event, probably through animals. I mean, one of the things that's really interesting when you look at the population density of the world is to realize how little undeveloped land there is in China, and how closely the Chinese population coexists with animals, which partially explains, I think, for why a lot of these viruses come from there.
Okay, that's great. The next question that we have is, how soon will we know if the Pfizer and Moderna vaccines protect against the new variants from South Africa, Brazil, and the UK? So how soon will we know and get an idea if we have to be doing a different vaccine?
I think the laboratory tests are out, and the laboratory test results say that they work just as well with the UK variant, but are less effective, but perhaps not tremendously so against the South African variant. Now, I may also say that the J&J vaccine underwent a clinical trial with one shot in South Africa when 90% of the circulating strains there were the South African variant. And though it was a little less effective in preventing mild disease for that strain, in preventing serious disease, which is not hospitalization, by the way, it was just as effective, 89%, as in the US, and it was 100% protective against that strain for preventing hospitalization and death. So at the same time, all these companies have started to make new variants against the South African strain because of abundance of caution, if you like. And the beautiful thing is, as Andre described, these RNA and DNA-based adenovirus-based vaccines, because they're giving the instructions rather than the protein, are very easy to adapt and make for new ones. So that's wonderful.
Yeah, and I would just point out that the goal of our vaccine was a 50% efficacy. We're a little bit spoiled because of the incredible efficacy of the vaccines that were generated. This 90 to 95% is just unheard of. And so, you know, if these were half as efficacious against the mutants as they are against the wild type virus, these are still very good vaccines.
Okay. The next question is about the length of the immunity from the new vaccines, is, you know, being still somewhat unknown. So what will happen if it turns out that, you know, immunity only lasts, say, 6 months? Do we run the risk of COVID sneaking back up on us even after vaccination seems complete? Or will we now have early warning that the immunity won't last long? And I guess along with that, we don't even quite even know those who have been infected seem to have some sort of immunity, but it doesn't seem to say for how long you've got the antibodies. So do we run the risk of COVID sneaking back up on us? And will we be smarter next time to be able to catch it, I guess, is the question.
Go ahead, Andre.
I think we won't know until enough time has passed for this to happen. I think there's been a large fixation on the antibody levels, which for some people seem to go down relatively quickly. But the cells that make those antibodies and the T cell response is also an important part of memory, and we won't know. We don't measure that part of it. So we don't really know how that's going to go. One of the issues with SARS-CoV-2 is that this was a new virus. None of us had immunity to it. And so this virus could pretty much just run like wildfire through the population. But it's also conceivable that as we get used to having multiple rounds of coronaviruses, and that may be what happens, it'll be like influenza or the common cold, that as a population will generate some what we call cross-reactive immunity. It's not perfect for the new virus, but it's okay. And that will suppress the really bad effects of these viruses. So who knows, really?
Yeah, that's one of the biggest challenges in understanding of immunology, where we have a hole, as to how a particular infection or a particular form of vaccine, how durable is the immune response that it generates. Although recent studies are showing that the memory B cells in people who have recovered from disease, this we don't yet know about those who are vaccinated, can really quickly turn on and ramp B cell antibody production up. As expected, by the way.
Okay, and then the next question is, can we target the viral genome packaging for treatment? Can we target the viral genome packaging for treatment?
I mean, I'll answer. I mean, of course we can. I think what we learned in the pandemic is that, you know, certain strategies we can mobilize very quickly, like the vaccine strategy, like the antibody strategy, and developing drugs to inhibit the virus. It's very difficult to do that in 6 months or a year. I mean, your typical drug takes 5 or 10 years to get through. And so that strategy, of course, could work. It's just a very slow, not able to be mobilized in the short time frame that we have.
Moreover, I think that, you know, if this becomes a regular problem, for example, like influenza or something like that, then I think there will be even more incentive to try and develop coronavirus-specific drugs. I mean, the ones that remdesivir, for example, just tries to block one of the steps in the replication cycle of RNA viruses. And this is a very generic thing. It was developed really for Ebola originally, and then was not licensed for that, and we have repurposed that. So I think if it becomes an endemic problem that we deal with every year, then companies like Andre's company will put money and time into trying to figure out a specifically designed treatment. But there is no, as I mentioned, there's no part of the viral replication cycle that is off bounds, including the packaging for a target for a drug.
Okay, so I don't see any other questions. And I mean, as I said, this has been really informative. And as you can tell from the questions, people were really looking for some great details here. As I mentioned, for those of you that weren't here, at the top of the talk, Viruses, Pandemics, and Immunity, this is out from MIT Press, and we have copies of it available at Labyrinth Book, who is our co-sponsor. They're a local independent bookstore here in Princeton, and they're going to give you a 10% discount. And I think, you know, the link is there in the chat if you want, and labyrinthbooks.com. And this has been the Princeton Public Library hosting this event, and I just really want to thank our two speakers tonight for the very thorough, and I loved all the different drawings and stuff, and so those are also included in the book. It's an illustrated book that's going to help you understand this in much better detail. And I want to thank you guys for working on this and getting it out to market so quickly, so that we can have a better guide to understanding what's going on in our world.
Thank you. Thanks for hosting.
Thank you.
Bye-bye.
It's been a great pleasure. And this will be going up on the library's YouTube channel if anybody wants to watch a replay or share it with friends. And I'll share the link out later on. But you can go to YouTube/PrincetonPL and see a history of some of our past talks. And so this will hopefully help educate other people as well, and let people know that this book is out there. And again, thank you to our sponsors, and I'll say good night.
Good night.
Good night.